Right now, inside every one of your cells, something invisible is either happening or not happening. If it is happening, your cells are clean, energized, and rebuilding themselves. If it is not happening, damaged proteins are piling up, worn out. Cellular machinery is cluttering the interior, and toxic waste is quietly accumulating. That accumulation does not stay quiet forever. It becomes inflammation, then disease, then accelerated aging. The process that prevents all of this has a name. It was awarded the Nobel Prize in Physiology in 2016. It is called autophagy, and today, you are going to learn exactly how to turn its switch on. The word autophagy comes from Greek roots meaning self-eating. That sounds alarming, but the reality is the opposite of harmful. When autophagy is activated, your cell builds a double membrane pouch called an autophagosome. This pouch engulfs damaged proteins, malfunctioning mitochondria, and other cellular debris. It then fuses with a lysosome, a specialized cellular organelle that acts like a molecular furnace, and burns the contents down to their raw materials. Those materials, amino acids, fatty acids, and sugars, are then recycled into new proteins and cellular structures. It is a perfectly designed circular economy. Inside every one of your 37 trillion cells, autophagy also functions as an immune mechanism, directly degrading bacteria and viruses that invade cells. When autophagy breaks down, the consequences are severe. Impaired autophagy is closely linked to the development of type 2 diabetes, cardiovascular disease, Parkinson's disease, Alzheimer's dementia, and multiple forms of cancer. The cellular switch that controls autophagy comes down to two key enzymes working against each other. The first is mTOR. When you eat and your blood glucose rises, insulin is secreted, and mTOR activates. An active mTOR tells your cells to grow and build, which means autophagy is switched off. The second enzyme is AMPK. When you are in a fasted state, when you exercise and deplete cellular energy, or when nutrients are scarce, AMPK activates. AMPK suppresses mTOR and directly triggers autophagy. The core principle behind every autophagy activation strategy is the same. Lower insulin, suppress mTOR, let AMPK take over. The timeline matters enormously. From the end of your last meal through roughly the first 4 hours, insulin and blood glucose remain high and mTOR is dominant. Between hours 4 and 12, glucose gradually drops and mTOR activity begins to fall. Past the 12-hour mark, AMPK begins actively switching on autophagy. Between 16 and 24 hours, autophagy flux is running at a meaningfully higher level. A landmark study on neuronal autophagy found that even short-term fasting produced a dramatic upregulation of autophagy in brain neurons, with measurable increases in autophagy markers after just 24 hours. The scientific consensus places peak autophagy efficiency somewhere in the 24 to 48-hour window, though significant benefits begin accumulating well before that, starting from the 16-hour mark. For practical purposes, this means a consistent 16-hour fast each day delivers real and measurable cellular cleaning benefits, without the risks that come with extreme multi-day fasting. The first autophagy switch is intermittent fasting. The most studied practical protocol is the 16-to-8 method, where eating is confined to an 8-hour window each day. If your last meal is at 8 p.m., and your first meal the next day is at noon, you hit 16 hours of fasting every single day. A 6-week alternate day fasting study found increased autophagy flux, alongside a significant reduction in myocardial infarct area. A study on prolonged intermittent fasting confirmed that autophagy increases in a time-dependent manner, while inflammasome activity and cellular aging markers simultaneously decrease. During the fasting window, black coffee and plain green tea are allowed and actually helpful. Black coffee suppresses mTOR and stimulates AMPK. Green tea catechins support autophagy activation. Milk, juice, sweetened beverages, and anything with calories will spike insulin and immediately turn the autophagy switch back off. The second autophagy switch is exercise. Exercise activates autophagy through a completely independent pathway from fasting, and the two work synergistically. When muscles contract, they burn through cellular energy reserves rapidly. That energy deficit activates AMPK, which suppresses mTOR and triggers autophagy. Research on high-intensity interval training found measurable changes in autophagy markers following acute bouts of exercise. A separate study confirmed that just 30 minutes of high-intensity exercise is sufficient to stimulate autophagy in healthy individuals. You do not need extreme exertion. Moderate-intensity aerobic exercise, brisk walking, cycling, swimming, and resistance training all activate autophagy. Exercising in a fasted state combines the fasting-induced and exercise-induced AMPK activation, simultaneously producing a compounded effect. 30 or more minutes of exercise at least 5 days per week is the most practical and sustainable way to keep your cellular cleaning system running consistently. The third autophagy switch is food. Compounds that mimic the cellular signals of fasting, even outside of fasting windows. Certain natural compounds suppress mTOR and activate autophagy directly. The most powerful of these is spermidine. Spermidine is a polyamine found abundantly in wheat germ, at approximately 24 to 35 mg per 100 grams, and in natto, at roughly 11 to 20 mg per 100 grams. A study published in a major life sciences journal confirmed that spermidine levels rise in both animals and humans during fasting and caloric restriction, and that this rise in spermidine is a key mediator of fasting-induced autophagy. Beans, tofu, mushrooms, and broccoli are also meaningful sources. The second key compound is resveratrol, found in red grapes, blueberries, and peanuts. Research has confirmed resveratrol directly induces autophagy by inhibiting the mTOR-ULK1 pathway and stimulating LC3 conversion, a marker of active autophagy. Third is quercetin, found most abundantly in capers, onions, apples, and broccoli. Quercetin activates AMPK and inhibits mTOR, creating overlapping effects with fasting. A review of natural autophagy activators concluded that curcumin, resveratrol, quercetin, and spermidine represent the most evidence-backed natural inducers of autophagy for combating age-related disease. Here are 3 practical recipes. The first is an autophagy morning smoothie. Blend a handful of frozen blueberries, a handful of spinach, 1 teaspoon of flaxseed, a 1/4 teaspoon of turmeric powder, and a small pinch of black pepper with water or unsweetened plant milk. The blueberries deliver resveratrol and anthocyanins. The spinach provides quercetin, turmeric adds curcumin, and flaxseed provides omega-3 fatty acids. Drink this within your eating window, not during the fasting window. The second is an autophagy lunch salad. Combine raw red onion, capers, steamed broccoli, and arugula, dressed with extra virgin olive oil and lemon juice. Onions and capers are the most concentrated food sources of quercetin. Broccoli supplies both spermidine and sulforaphane, and olive oil contributes oleuropein, which also supports autophagy pathways. The third is an autophagy dinner soup. Simmer doenjang or miso paste broth with firm tofu, shiitake mushrooms, bok choy, and garlic. Tofu and fermented soy paste are excellent spermidine sources. Shiitake mushrooms are rich in polyamines, and garlic provides anti-inflammatory and autophagy-supporting compounds. Important safety notes are essential. Anyone taking diabetes medication or insulin, pregnant or breastfeeding individuals, adolescents, and anyone with a history of eating disorders should consult a doctor before attempting extended fasting. Cancer patients should not independently undertake aggressive autophagy protocols, as autophagy plays a dual role in cancer biology, and its effect in advanced disease differs from its preventive role in healthy tissue. Extreme fasting or severe caloric restriction can reduce muscle mass and lower basal metabolic rate, so any fasting practice should be paired with adequate protein intake and strength training. Finally, autophagy operates at a cellular level, and meaningful changes in how you feel, including better skin, more stable energy, improved cognitive clarity, and reduced inflammation. Markers accumulate over weeks to months, not days. Consistency matters far more than intensity. Autophagy is not a trend. It is one of the most fundamental survival mechanisms your cells possess. 5 switches activate it: 16-hour intermittent fasting, fasted state exercise, spermidine and quercetin and resveratrol-rich foods, 7 to 8 hours of deep sleep, and gradual cold exposure. You do not need to activate all 5 at once. Start with the one that fits your life most easily today, and add the others one by one. When your cells are clean, everything downstream in your body works better. That is not a marketing claim. It is what the 2016 Nobel Prize was given for discovering.